Mitosis or mitotic cell division is a type of cell division that's going to result in two genetically identical cells. So they're going to be exact copies. So if you start with a diploid cell, it's going to make two diploid cells that are exactly the same as the mother. So we tend to use the feminine with cells, so mother and daughter. So the two daughters will be exactly the same as the mother. But mitosis can take any cell and make a copy of that cell. So you can have a haploid cell, and same thing. You can copy it and make the exact same haploid cell. So making more of the same. You could take a triploid cell, cells with 3 copies of chromosomes, and you could make 2 cells that were triploid. So it doesn't matter what you start with, mitosis is always just going to make 2 of the exact same. And depending on the organism, you'll see mitosis carrying this out. So again, remember that there's really 2 steps to cell division. There can be the division of the nucleus and cytokinesis, which is the division into 2 cells. Some types of cells will never do this. So you'll find out in A&P that muscles, for example, will divide the nucleus, but not the cytoplasm. So what role does mitosis have in cells? Well, for some cells, it's reproduction. So for organisms that asexually reproduce, that means reproduction without sex, mitosis can just take 1 cell and make 2 new cells. So, for example, an amoeba, which is what's in this example, is dividing to make 2 new amoebas. So the mother amoeba becomes her 2 daughter amoebas. In organisms like ourselves that are multicellular, mitosis is important for growth and development. So this is how we get bigger, taller, fatter, is through mitosis. It's also part of development. So when you go from being a fertilized egg to being an embryo and to eventually an adult, that process is involving mitosis. Inside of our bodies, we're always renewing our tissues, and so mitosis plays a role in that. In this example, this is making new blood cells, but your skin is also constantly doing this. Places like your intestinal lining are carrying out lots of mitosis to replace the cells. So again, mitosis is going to divide the cell and the DNA, and it's really important that it sorts the chromosomes so that it gets the exact same number of chromosomes into the daughters as were present in the mother. So it's really an elaborate sorting of the chromosomes. And in order to sort the chromosomes, it's going to use something called the spindle apparatus, or the spindle for short. The spindle is going to be what grabs onto the chromosomes and sorts them and moves them around. So before we get into the phases of mitosis, we'll just look at an example of the spindle. This is the metaphase spindle. So this is what the spindle looks like at metaphase, but it lets us see all the key parts of it. So the mitotic spindle has 2 sides to it, or 2 poles, kind of like the North Pole and the South Pole. At each pole you have a centrosome. So here are the centrosomes, right? That's that term there if you want to write it down. So the 2 centrosomes, and out of those centrosomes grows microtubules. So the microtubules grow out of the centrosome. So I'm going to grow some microtubules here. So the ones I've drawn in red are kinetochore microtubules. Kinetochore microtubules will grow from the centrosome and attach to the chromatids. Astral microtubules, I'm going to draw in blue, are going back from the centrosome, so notice it's going the other direction, and holding the centrosome in position. So they attach to the plasma membrane and hold the centrosome in position. So they position the spindle in the cell. And then I'll do green for the last kind. These are called polar microtubules, and what they're going to do is push against each other. So this one and this one are coming from opposite ends. They can push against each other, and as they push against each other, they're going to stretch the cell. So as they push against each other, they'll take a cell that looks like this, so here's them pushing against each other, right? Force this direction, force this direction. They're going to push against each other, and that's going to slowly lengthen the cell. So the cell starts out as round, it becomes more elongated as the polar microtubules separate the two poles. So the centrosomes get pushed further apart from each other by the polar microtubules. So that's our basic spindle, and we're going to see the spindle doing stuff during mitosis. So remember that mitosis is broken into 5 phases: prophase, prometaphase, metaphase, anaphase, and telophase, or as most people, or many people remember it, PP-MAT. I don't know, that usually just sticks in people's heads just fine. PP-MAT. Prophase, prometaphase, metaphase, anaphase, and telophase. So when we go into prophase, the DNA is already copied. So remember that G1, S, and G2 have already happened. And the DNA was copied during the S phase. G1, S, and G2 are collectively called interphase. So the DNA was already replicated during interphase. So in the example cell that we're going to be looking at, there was 6 chromosomes. So now that it's replicated, there's 12 chromatids. They're still joined together at the centromere. At the beginning of, so in prophase, the DNA is already copied. So what begins to change is that the DNA will condense. So the chromosomes become much more visible. So we talked about this when we were talking about DNA. DNA can be very loose or it can be very tight. During interphase, the DNA is very loose. The very first thing that happens in prophase is that DNA becomes very condensed, and when you're looking in the microscope, it's going to look much more noticeable. At the same time, the nuclear membrane begins to dissolve. So you'll see that when you look in, for example, the microscope, you'll see the little membrane kind of around the DNA. What will happen is that will disappear, and the DNA will be kind of all over the cell. So then it goes into prometaphase. The mitotic spindle is fully formed at this point. You can't see the spindle under our microscopes. So I want to let you know that because you're going to be using the scopes to identify the stages, but you can't ever see the spindle with the light microscope. So there's not, in the microscopes, we're not going to see a lot of this happening, but the centrosomes have moved apart the 2 poles at this point. The spindle fibers are interacting with the sister chromatids, and when you watch the video of mitosis, you're going to see that what's happening is that the chromosomes are going to be getting moved around. So they're attached, but they're going to be getting moved around. And I'm just going to ignore the phone here so that I can keep going with this video. So the chromosomes are going to get moved around during this phase, but you won't see that in a fixed cell. So this is what this looks like. This is interphase. And so what we see in interphase is there's just microtubules all through the cell. This is the DNA. It's very loose. When the cell goes into prophase, the first thing that happens is that DNA gets really condensed. And so you can see that here. What was sort of light dispersed blue becomes dark purple, and each one of those purple things is the chromosomes. And you can see that in the diagram as well. The centrosomes are beginning to separate, which this is not using a light microscope, so you can see the microtubules beginning to form out of the centrosomes. Prometaphase, the spindles are fully formed and separated, so you can see this is one centrosome, this is the other centrosome, and here's the microtubules. The DNA is very compact, the nuclear membrane is gone, so see how this is still a circle? The nuclear membrane is still there. Now the nuclear membrane is totally gone, so it's not being held in that circle anymore. So these chromosomes are going to get moved around, and as they get moved around, they're eventually going to get to metaphase. So metaphase is when the chromatids are going to line up in the center of the cell. When the alignment's complete, the cell is sort of at metaphase, and that sets up the cell to go into anaphase. So once they're lined up, anaphase is when the chromatids will separate. So each chromatid will separate from its partner, and then they become individual chromosomes. The kinetochore microtubules will shorten and pull the chromatid, or now the new chromosome, towards its pole. And then telophase is when the centrosomes, or sorry, rather the chromosomes get to their side of the cell, and then the nuclear membrane will reform. So that's what these look like. So in metaphase, our chromosomes are going to be lined up down the center. So they'll get moved around until they line up down the center. This is kind of a very special moment that the cell recognizes because it has an internal sort of sense of something called the metaphase plate. So the metaphase plate runs down the center, and so when those chromosomes get aligned on the metaphase plate, that will stimulate separation of the chromatids. So here now the chromosomes or the chromatids have separated and become individual chromosomes. So you can see these are now heading to this side and these are now heading to this side. And then telophase is when the chromosomes are all the way over to their own side. A nuclear membrane is now reforming around them, so we're making 2 new nuclei. And then at the same time, and really these are happening together, telophase and cytokinesis, the cytoplasm begins to separate. In the case of animals, it'll separate with a cleavage furrow that literally squeezes the 2 cells apart. Imagine squeezing a balloon into 2. And at the end of telophase and cytokinesis, we're going to have 2 new cells that then go into the cell cycle independently and start over at G1. So the cleavage furrow I mentioned already. So you can see this is a cell in cytokinesis, and the cleavage furrow is literally pulling across it like a drawstring. It's going to separate it into 2 new cells. Plants can't do cytokinesis that way because of the cell wall. So what you see in plants, this is the original cell. Telophase is done. I can tell telophase is done because there's 2 new nuclei here. But what is happening is this faint line down the middle, that faint pink line, is called a cell plate. A cell plate forms between the 2 nuclei, and then it will eventually become a new cell wall. So first it's a cell plate. Notice it doesn't go all the way to the edges, and it's not as thick as a cell wall, but it will eventually become a cell wall. And so for plants, that's how they'll become 2 new cytoplasms. So to recap, mitosis is prophase, prometaphase, metaphase, anaphase, telophase. In prophase, the nuclear membrane will come apart, the chromosomes will condense. In prometaphase, the chromosomes will be attached to the spindle and moved around until they reach the metaphase plate. That puts us into metaphase. When those chromosomes are lined up down the middle in the metaphase plate, that will stimulate anaphase, which is the separation of the chromosomes. The chromosomes move to either side of the cell, so each pole. And then when those chromosomes reach the edge of their side, they'll reform a nucleus. And so now you have 2 new nuclei. And cytokinesis, either by a cleavage furrow or a cell plate, will turn that into 2 new cells. And so that's how we get from 1 cell to 2 cells.